Alright, well, I'm sure you recognize this is the electron transport chain. We're going to start talking about this in a little bit more detail, but first, introduction, the big picture. We should recognize that the ETC is used to create chemical energy in the form of ATP, and it couples an electron donor, and if you remember the NADH and the FADH2, these are the reducing equivalents, and so it couples electron donors with electron acceptors, and the transfer of hydrogen ions across the membrane creates an electrochemical gradient. Just the big picture here. I could say creates, it actually is mediated by, or caused by, an electrochemical gradient, and the gradient allows the transfer. ATP synthesis is powered by the flow of hydrogen ions back into the mitochondrial matrix. So remember that the electrons moving across the membrane creates the energy we need to make ATP, and so ATP synthase is used in this process, and we can see this in the next slide. So let's start talking about it. And continuing on, let's talk about the mechanisms of oxidative phosphorylation. Just the basics here, folks. So remember that the mitochondria is like the nucleus in that it has two membranes. And what we're doing here quite simply is creating a polarity, a charge difference in hydrogen ions, protons, between the inside and the outside, or the inner membrane here. And so the membrane here is permeable to very small molecules and ions, it is essential for producing electrochemical gradient that drives ATP synthesis. A controlled influx of protons, which are the hydrogen ions, through ATP synthase produces the ATP, and we see that in the ATP synthase area, and we're going to talk about that more in the next slide. But basically this is a hydroelectric power plant, and we are sending these hydrogen ions through the protein pumps, these transmembrane protein channels, and there are three basically that move, and they are powered by electrons, as you know, at the reducing equivalents of NADH and FADH2, and we're going to see how that works in the next slide. But we need to recognize here that the process is quite simple, and that finally the store of hydrogen ions, the protons, it creates a polarity in that it's much more above than it is below, and that it drives into the ATP synthase into this channel, this protein channel, which creates ATP, and we're going to see the details about that in the future slide. So now let's turn our attention to the Tim and Tom complex down at the bottom. You can see it says large nuclear encoded proteins. What's happening here, for example, let's look at Tom. Tom, it stands for transporter of the outer membrane, and what this is doing is it is importing channel transmembrane proteins, and the Tim is the transporter to the inner membrane. So this Tim-Tom complex describes the translocation of proteins produced in the nuclear DNA, and they are used here. They're sent through the mitochondrial membrane for use in oxidative phosphorylation. And as we were talking about before, remember that the mitochondrion does make its own proteins and has ribosomes. So about 15% of the protein is made inside, and 85% is brought in. This is an important thing to know. But let's remember that the mitochondrion does have, if you look in the middle, you can see that the mitochondrion does have a place where it can make DNA. It has DNA, and that it can make its own ribosomes, which makes its own proteins. So the take-home message here is that the mitochondria does have its own DNA and does have its own messenger RNA, which is going to make these proteins, and then about 15% of the proteins are made inside and 85% are brought in.